Okay, welcome back. You're ready for another lecture. That's a good sign. And hopefully you've gotten a thorough explanation and understanding about amino acids because as you see, it's not just amino acids but also proteins that we'll discuss in this chapter. And in this third lecture here, we're getting ready to talk about those proteins. So in order to make those proteins that are very important, as we started off our whole discussion of this chapter in the very first lecture, saying how varied and valuable the protein function is, well, we got to connect these amino acids. The amino acids are fascinating. Hopefully you've enjoyed everything up until now, but until we start linking them together, they're just amino acids. So in order to make those proteins, we're going to have to make some peptide bonds. And again, the biologists still like that term. The chemists, most of us prefer the term amide bonds, right? We talked about amides there in the previous chapter, and that's what the peptide bond is. It's an amide. It's not anything special. It's just different because it's biological, whereas we can have amides that are non-biological. Okay, so first and foremost, a peptide bond is an amide bond, right? It forms between the carboxyl group of one amino acid and the amino group of the next amino acid. It would contain a nitrogen, a free protonated ammonium terminus written on the left by convention. It also contains a carboxylic acid group, a free carboxylate terminal on the right by convention, and there you see it. So that's our formation of glycylalanine by taking glycine and having its carboxyl group interact with alanine's protonated ammonium group, and we form that glycylalanine amide linkage. So that's our peptide, right? Joining two amino acids into a single bi-amino acid there. Also, of course, water as a coproduct. Okay, and here we do it with representations more like ball and stick models, so you can see what's happening there. And again, we make a dipeptide out of those two single peptides, single amino acids, a dipeptide because we have one peptide linkage or one amide bond there, combining the glycine with the alanine. And of course, don't forget the water, that's our necessary coproduct. Now, just because we're in the biological chemistry end of things doesn't mean we can forget about naming. And of course, we had a name for that dipeptide that we showed on the previous slides. So what you probably noticed is that a dipeptide is named with a YL ending for the N-terminal, the free protonated ammonium amino acid, and then the full amino acid name of the free carboxyl or carboxylate at the pi at the C terminus. So there we have it. Because we had alanine in our original amino acid, the alanine is what used its carboxylate to interact with glycine's protonated ammonium. We would have, again, in this case, it's a tripeptide, so we have another serine residue that has its protonated ammonium interact with that glycine's carboxylate. So we would name this thing as alaninyl glycylserine. So whenever we're dealing with something that doesn't have the carboxyl end free, it gets that YL ending. So because we have the carboxylic acid portion joining to form peptide bonds from the alanine and the glycine, whereas serine has that free deprotonated carboxylic acid, we have to take the roots from alanine and glycine and add that YL ending, and then serine gets to have its whole name, and that's our typical pattern. We can also abbreviate it. We have 3-letter abbreviations and 1-letter abbreviations, and sometimes they're logical, like ALA for alanine, GLY for glycine, and SER for serine, or AGS is the 1-letter abbreviations. Sometimes they make almost no sense, as you'll see in a little while. Okay, so let's review how we would draw a peptide now that we've reviewed how we would name a peptide. So first we would draw the structures for each of those individual amino acids involved in the peptide, starting with the N-terminal amino acid on the extreme left. we'd remove the O atom from the carboxylate group of the N-terminal amino acid and 2 of the hydrogens from the adjacent amino acid. And that's how we're going to get our water, right? That's our coproduct for each of those peptide or amide bonds we form. So you'd repeat the process until you get to the C-terminal amino acid, and then finally connect the remaining parts of the amino acids by forming those amide or peptide bonds. Okay, so let's see how you do with that. In this learning check, we're going to test your ability to draw and name. So for this one, you're asked to draw the condensed structural formula, or of course the skeletal structure if you prefer, and give the name and abbreviation for the dipeptide. And I won't say it because you're going to name it, so SER-THR. So there you have it. Clearly there's serine and threonine residues, but I'll leave you to the naming and to drawing the structure of that dipeptide. Stop the video here while you do that. Good luck. Okay, so there we have it. We have the serine, which is retaining its free amino or ammonium terminus in this case. We have the threonine, which is going to have its free carboxyl terminus. And so there we have the peptide bond that we form by taking that O minus and 2 H's, one of which would be a plus to neutralize that O minus. That's how we get our water. That's how we make our peptide or amide bond. The name, therefore, would be cerylthreonine, or just SER-THR for the 3-letter abbreviations. Hopefully that's what you have, or at least now you understand where we were going with it, and you can practice on your own. If you're really stuck and the additional practice is just frustrating you even more, then please do reach out to me for some additional guidance. Okay, so let's see how well you're getting all this. In this learning check, you're asked to write the names and 3-letter abbreviations of the tripeptides that could form from 2 glycines and 1 alanine. Be careful that they're unique. You can form at least 2 different possibilities, right? So go ahead and see what you can make from those residues, and stop the video while you do that, and then start it back up when you're ready to check your work. Good luck. Okay, so here we have it. So you could have the 2 glycines come first and the 1 alanine, right? Just like we sort of ended that learning check itself. And so that would be glycylalanine. You could have the glycines at the end and the alanine in the middle for glycylalaninyl glycine. Or you could start with the alanine and then end with the 2 glycines to form alanyl glycyl glycine. So there you have it. Those are the 3 possibilities. Anything else you do is just one of those possibilities over again. It would not have a unique name. You don't even have to draw the structures if you're comfortable with this, but if you did do the structures and you want to run them by me, then certainly email me a picture of them and I can weigh in on that. But if it's all making sense and you could do it just from the names and 3-letter abbreviations, then that's good. If you could catch these 3 different variants and that there are no other possibilities, then you're in really good shape. And if you're not there yet, please do reach out for some help. Okay, let's do another learning check just to make sure everybody's there. Even if you think you're there, let's confirm that. So now we're asked, what are the possible tripeptides formed from 1 each of leucine, glycine, and alanine? So now things are a little more complicated because we don't have any duplicates. So you can just do the 3-letter abbreviations. If you really love the naming, you can name them all as well, but at least the 3-letter abbreviations for the possible tripeptides from these 3 unique amino acids. Okay, so this is where you can use your old probability things from math, right? Any one could be the start. So we could have, let's say, leucine as the start because that's how it was proposed to you in the original learning check. So leucine, glycine, and alanine, that's how we gave them in the original. So why not start there? The other thing we could do is keeping leucine at the front. We could have leucine, then alanine, then glycine. That's all we can do with starting with leucine, right? Then we could look at our possibilities starting with alanine. So alanine, leucine, glycine, or alanine, glycine, leucine. Finally, let's start with glycine, and we could go glycine, alanine, leucine, or glycine, leucine, alanine. So there we have it. From those 3 different amino acids, we can make 6 different tripeptides. If you remember your probability from math, since any one of the 3 could start it, and then any 2 of the remaining could be the middle, and then obviously that just leaves 1 for the end. If you remember your probabilities there, you could have 3 times 2 times 1. You would have been able to predict that you would have 6 total as a possibility. But if that's unpleasant memories from middle school or high school math or whenever you last did that stuff, then certainly just logic and puzzle work here works just as well. But there we have it, 6 possibilities from those 3 unique amino acids. Okay, and here it is, the last learning check for this lecture, I promise. In this case, you're asked to write the name and 3-letter abbreviation for the following tetrapeptide. So there's 4 amino acids there. This will give you a good chance to see how well you remember the names based on the side chains. And certainly if you need to peek, at this point we're still early on, so that's acceptable, but you should really be striving to get those side chains committed to memory and the names as well, because they're quite important. There's only 20 of them, right? Easy for me to say, huh? So go ahead, stop the video here while you figure out this tetrapeptide and start back up when you're ready to check your work. Good luck. Okay, hopefully you were able to arrive at ALA-LEU-CYS-MET, or alanyl leucyl cysteal methionine, or something like that, right? I think I pronounced that correctly. But there we have it. So alanine, right, the R group is the methyl group. So we've used that example quite a bit. Hopefully that was an easy one. Leucine we haven't talked a whole lot about if you needed to peek for that one, and also distinguish it from isoleucine. At this point, of course, I'm not too concerned about that, but hopefully you'll be able to make that distinction. Cystine's another big one because of that thiol group that's available to make those disulfide bonds. So I mentioned earlier that cystine's one we should know and associate with those disulfide bridges. So hopefully you have that one committed to memory. And then methionine just happens to be the other sulfur-containing amino acid. So hopefully you have already caught on there as well. So there we have it, identifying the 4 amino acids that make up that tetrapeptide. Hopefully that went well. If it's frustrating you and things just aren't getting better with practice, please don't just spin your wheels without any benefit. Reach out to me. I'll try to point you in a direction that is much better suited for getting you where you need to be with this content. Otherwise, if things are going well, then that's great, and we'll meet again in the next lecture for this chapter.